Welcome to the fascinating world of microbial genetics. In this video, we'll explore how microbes such as bacteria and archaea use genetic coding to survive, adapt, and thrive in diverse environments. Microbes are among the oldest and most adaptable life forms on Earth. Their genetic material, often organized in a single circular chromosome, holds the instructions for all their functions. Understanding genetic coding in microbes not only reveals the secrets of their survival, but also helps us harness their abilities for medicine, industry, and environmental solutions. Let's embark on this journey to decode the language of life at its smallest scale. Genetic coding refers to the way living organisms store and transmit information using DNA or RNA. In microbes, this code is a sequence of nucleotides adenine, thymine, cytosine, and guanine that form genes. These genes are like sentences in a book, each instructing the microbe on how to build proteins, replicate, and respond to its environment. The genetic code is universal, meaning the same code is used by almost all living things, but microbes have unique ways of organizing and expressing their genes, making them incredibly versatile. Most microbes have their genetic material in the form of a single circular DNA molecule. This structure is compact and efficient, allowing microbes to quickly replicate and adapt. Some microbes also carry small, extra pieces of DNA called plasmids, which can provide additional traits like antibiotic resistance. The simplicity of microbial DNA structure is one reason why they can evolve rapidly and survive in extreme conditions. This unique organization sets microbes apart from more complex organisms, whose DNA is often linear and stored in a nucleus. Central dogma of molecular biology describes how genetic information flows from DNA to RNA to protein. In microbes, this process is streamlined for efficiency. DNA is first transcribed into messenger RNA, mRNA, which then serves as a template for building proteins. These proteins carry out essential functions from metabolism to defense. Because microbes often live in changing environments, their genetic coding and expression systems are highly responsive, allowing them to quickly adjust to new challenges. Gene expression in microbes is tightly regulated. Not all genes are active at all times. Microbes use regulatory sequences in proteins to turn genes on or off depending on their needs. For example, when nutrients are scarce, certain genes may be activated to help the microbe survive. This flexibility is key to microbial success. By controlling gene expression, microbes can conserve energy and resources, ensuring they only produce what is necessary for survival. One unique feature of microbial genetics is the operon. An operon is a cluster of genes under the control of a single promoter. This arrangement allows microbes to coordinate the expression of related genes, making their responses more efficient. For example, the lac operon in E. coli controls the breakdown of lactose. When lactose is present, the operon is activated and the necessary enzymes are produced. This system helps microbes quickly adapt to changes in their environment. Mutations are changes in the genetic code. In microbes, mutations can occur spontaneously or be induced by environmental factors. While some mutations are harmful, others can provide advantages, such as resistance to antibiotics. Because microbes reproduce quickly, beneficial mutations can spread rapidly through a population. This process drives microbial evolution, allowing them to adapt to new threats and opportunities. Understanding mutations helps us track the spread of diseases and develop new treatments. Like most higher organisms, microbes can exchange genetic material with each other through horizontal gene transfer. This process allows them to acquire new traits rapidly, such as antibiotic resistance or the ability to metabolize new substances. There are several mechanisms for horizontal gene transfer, including transformation, transduction, and conjugation. This genetic flexibility is a major reason why microbes are so adaptable and can thrive in diverse environments. Transformation is one way microbes can take up genetic material from their environment. In this process, a microbe absorbs free DNA fragments released by other cells. If the new DNA is compatible, it can integrate into the microbe's genome, providing new abilities. This mechanism is especially important in natural environments, where microbes are constantly exposed to genetic material from many sources. Transformation contributes to genetic diversity and rapid adaptation. Transduction is another form of horizontal gene transfer, where viruses called bacteriophages transfer genetic material between microbes. When a bacteriophage infects a microbe, it can accidentally package some of the host's DNA and deliver it to another cell. This process can spread beneficial genes quickly through a population. Transduction highlights the interconnectedness of microbes and viruses in shaping genetic diversity. Conjugation is often described as microbial mating. In this process, two microbes connect through a structure called a pilus and transfer genetic material directly. This exchange often involves plasmids, which can carry important traits like antibiotic resistance. Conjugation allows microbes to rapidly share beneficial genes, making populations more resilient and adaptable. It's a key driver of genetic innovation in microbial communities. Plasmids are small, circular DNA molecules found in many microbes. Unlike the main chromosome, plasmids can replicate independently and often carry genes that provide advantages, such as antibiotic resistance or the ability to metabolize unusual substances. Plasmids can be transferred between microbes through conjugation, making them powerful tools for adaptation and survival. Scientists also use plasmids in genetic engineering to introduce new traits into microbes. One of the most significant consequences of microbial genetic coding is the spread of antibiotic resistance. Genes that confer resistance can be located on plasmids or chromosomes and are easily shared through horizontal gene transfer. This rapid spread makes treating infections more challenging. Understanding how microbes acquire and share resistance genes is crucial for developing new strategies to combat antibiotic-resistant infection. CRISPR is a genetic system found in many microbes that acts as an immune defense against viruses. When a microbe survives a viral attack, it stores a piece of the virus's DNA in its own genome. If the virus attacks again, the microbe uses this stored information to recognize and destroy the invader. CRISPR has revolutionized genetic engineering, allowing scientists to edit genes with unprecedented precision. Scientists have harnessed the power of microbial genetics to engineer microbes for beneficial purposes. By inserting, deleting, or modifying genes, researchers can create microbes that produce medicines, clean up pollution, or generate renewable energy. Genetic engineering relies on our understanding of microbial genetic coding and the tools microbes naturally use to exchange and modify DNA. Microbial genomics is the study of the entire genetic material of microbes. Advances in DNA sequencing technology have made it possible to read and analyze microbial genomes quickly and affordably. This field has revealed the incredible diversity and adaptability of microbes, uncovering new genes, pathways, and potential applications in health, agriculture, and industry. Microbial genetic coding has transformed medicine. Engineered microbes are used to produce life-saving drugs like insulin and antibiotics. Understanding microbial genetics also helps us track and control infectious diseases, develop vaccines, and design targeted therapies. The study of microbial genomes continues to open new frontiers in medical science. Microbes are essential in industrial biotechnology. By manipulating their genetic code, scientists can create microbes that produce biofuels, break down waste, or synthesize valuable chemicals. These applications are more sustainable and environmentally friendly than traditional methods. Microbial genetics is at the heart of many green technologies shaping our future. Microbial genetics plays a crucial role in environmental science. Engineered microbes can clean up oil spills, degrade plastics, and remove toxins from soil and water. By understanding and modifying their genetic code, we can harness microbes to solve some of the world's most pressing environmental challenges. These solutions are often more effective and less invasive than chemical methods. In agriculture, microbes with engineered genetic codes are used to promote plant growth, protect crops from pests, and improve soil health. These beneficial microbes reduce the need for chemical fertilizers and pesticides, making farming more sustainable. Advances in microbial genetics are helping to feed a growing global population while protecting the environment. Microbes are incredibly diverse, with millions of species adapted to every environment on Earth. Their genetic coding reflects this diversity, with unique genes and pathways that allow them to survive in extreme heat, cold, acidity, or salinity. Studying microbial diversity helps us understand the full potential of life and discover new genes with valuable properties. Metagenomics is the study of genetic material recovered directly from environmental samples. This approach allows scientists to analyze entire microbial communities without needing to culture individual species. Metagenomics has revealed vast, previously unknown diversity and has important applications in ecology, medicine, and biotechnology. Synthetic biology combines engineering and biology to design and construct new biological parts and systems. In microbes, synthetic biology enables the creation of custom genetic circuits that perform specific tasks, such as producing biofuels or detecting toxins. This field is rapidly advancing, opening up new possibilities for innovation and problem-solving. Microbes communicate with each other using chemical signals in a process called quorum sensing. This allows them to coordinate behavior, such as forming biofilms or launching attacks on host organisms. Quorum sensing is controlled by genetic circuits that respond to population density, making microbial communities highly organized and responsive. Biofilms are structured communities of microbes that stick to surfaces and to each other. The formation of biofilms is controlled by genetic coding and communication between cells. Biofilms can be found in natural environments, industrial settings, and even inside the human body. They are often more resistant to antibiotics and environmental stress, posing challenges and opportunities for science and medicine. Microbes cause disease by using their genetic coding to produce toxins or evade the immune system. Pathogenicity genes can be located on chromosomes or plasmids and are often transferred between microbes. Understanding the genetic basis of pathogenicity helps us develop better diagnostics, treatments, and preventive measures against infectious diseases. Microbes have evolved a variety of genetic mechanisms to resist antibiotics and other threats. These include modifying drug targets, pumping out toxins, and breaking down harmful substances. Resistance genes can spread rapidly through populations, making it essential to monitor and manage their use in medicine and agriculture. Mobile genetic elements such as transposons and integrins are DNA sequences that can move within and between genomes. These elements play a major role in microbial evolution by spreading genes, including those for antibiotic resistance. Mobile genetic elements contribute to the dynamic nature of microbial genomes. Microbes use complex networks of genes and regulatory elements to control their functions. These networks allow microbes to sense their environment and adjust their behavior accordingly. By studying gene regulation, scientists can better understand how microbes respond to stress, interact with hosts, and carry out specialized tasks. Epigenetics refers to changes in gene expression that do not involve alterations to the DNA sequence itself. In microbes, epigenetic mechanisms include DNA methylation and modification of proteins associated with DNA. These changes can affect how genes are turned on or off, influencing microbial behavior and adaptation. Microbes often face stressful conditions such as changes in temperature, pH, or nutrient availability. Their genetic coding allows them to sense and respond to these stresses by activating specific genes. This adaptability is a key reason why microbes can survive in extreme environments from hot springs to deep-sea vents. Many microbes live in close association with other organisms, forming symbiotic relationships. Genetic coding enables microbes to interact with their hosts, exchange nutrients, and provide benefits such as digestion or protection from pathogens. Symbiosis is a driving force in evolution and ecosystem health. Genomic islands are large segments of DNA acquired from other organisms, often through horizontal gene transfer. These regions can carry genes for antibiotic resistance, pathogenicity, or metabolic functions. Genomic islands contribute to the rapid evolution and specialization of microbes. Genome editing tools such as CRISPR-Cas systems allow scientists to precisely modify microbial DNA. These technologies are used to study gene function, create new traits, and develop microbes for industrial and medical applications. Genome editing is revolutionizing our ability to harness microbial potential. Phylogenetics is the study of evolutionary relationships among organisms. By comparing genetic sequences, scientists can trace the ancestry of microbes and understand how they have diversified over time. Phylogenetic analysis helps identify new species and track the spread of pathogens. Population genetics examines the distribution of genetic variation within and between microbial populations. This field helps us understand how microbes adapt to their environments, spread resistance genes, and evolve new traits. Population genetics is essential for managing infectious diseases and conserving microbial diversity. Epistasis refers to interactions between different genes that affect a microbe's traits. In microbes, epistasis can influence how genetic changes impact fitness, adaptation, and evolution. Understanding these interactions helps scientists predict the outcomes of genetic modifications and natural mutations. Genetic drift is a random process that changes the frequency of genes in a population over time. In small microbial populations, drift can have a significant impact, leading to the loss or fixation of certain genes. Genetic drift contributes to the diversity and evolution of microbial communities. A genetic bottleneck occurs when a population is drastically reduced in size, causing a loss of genetic diversity. In microbes, bottlenecks can happen during infection, transmission, or environmental changes. Bottlenecks influence the evolution and adaptability of microbial populations. Genetic recombination is the process by which microbes exchange genetic material, creating new combinations of genes. This process increases genetic diversity and can lead to the emergence of new traits. Recombination is a key driver of microbial evolution and adaptation. Some microbes, especially those living in stable environments or as parasites, undergo genome reduction. This means they lose unnecessary genes over time, streamlining their genetic code for efficiency. Genome reduction can reveal which genes are essential for survival and adaptation. Genetic redundancy occurs when multiple genes perform similar functions. In microbes, redundancy can provide a backup system, ensuring survival if one gene is lost or damaged. This feature contributes to the resilience and adaptability of microbial populations. As our ability to manipulate microbial genetics grows, so do ethical considerations. Issues include the potential for unintended consequences, environmental impact, and the responsible use of engineered microbes. Ongoing dialogue among scientists, policymakers, and the public is essential to ensure safe and beneficial applications. The field of microbial genetics is rapidly advancing. New technologies such as single-cell genomics and advanced gene editing are expanding our understanding and capabilities. The future holds exciting possibilities for medicine, industry, and environmental sustainability, driven by continued exploration of microbial genetic coding. Summary: genetic coding in microbes is a cornerstone of life's adaptability and innovation. From basic survival to cutting-edge biotechnology, microbes use their genetic code to shape the world around us. By studying and harnessing microbial genetics, we unlock new solutions for health, industry, and the environment. The story of microbial genetics is far from over, and its potential continues to inspire scientific discovery.